the beginning, and indeed, our journey on the serine proteases is a fairly long one. It started off almost as soon as our lab began in 1998, and amazingly, it's still going on. Okay, the first story is going to be on looking at serine proteases in two different genomes, two different plant genomes, and this is going to be presented by Lokesh, who's here with us online. And he is, in fact, going to explain how we kind of compared and first of all searched and compared serine proteases in two different plant genomes, one representing the monocot, another representing the dicot, and relatively well characterized, namely Arabidopsis and rice. And in fact, there are as many as 50 different families, subfamilies of serine proteases, and he's going to talk about them. Briefly, what's interesting, from my viewpoint, is that, of course, each time we perform genome-wide survey, the experience is very different, and the standardization has to be very system-specific, very family-specific. However, what we were amazed is, whereas there are as many as 40% of the genomes of the serine protease across these two plant genomes were the best homologs, and that is normal. Many of them turned out to be atypical, bearing atypical members, and this happened for deg, clip, lawn, rhomboid proteases. Whereas there were species-specific members observed for a couple of serine proteases, namely the sublysine and serine carboxypeptidase. And finally, certain families of serine proteases like the deg-P, Prolyloligopeptidases, which you will hear much later, the clip protease and the rhomboids, in fact, had higher amount of orthology between the two highly well separated in evolutionary scale, namely the monocot and the dicots, and there seems to be appreciable amount of orthology between them. This is going to be presented by Lokesh as first story, and in fact, he's also going to tell how the phylogeny kind of yields and separates those kind of family members which have a certain type of domain architectures. And about the domain architectures, we had discussed this in the previous talks. I don't have to explain it to you. Okay, that is the first story. And then second story is on our kind of worrying about the functions, the biological functions of the serine proteases, namely the trypsin type serine proteases. And in fact, this problem was introduced to us as early as 1999 by late Professor Veronica Rodriguez of the TFR Mumbai. And she pointed out in the Drosophila genome, an appreciable amount of the annotated trypsin type serine proteases were not having the functional residues, namely the catalytic triad, as you can see in the middle, as his sensor. And such residues are vitally important, and whereas there were a number of them which do have the functional residues intact, there were also appreciable amounts of them which had at least one of the catalytic site residues totally missing. And the question really is, knowing that they are not silent and they are being expressed, what is the overall biological role of such non-functional serine proteases? That's the second part A, or the part A of the second story, which Lokesh is going to present. And this we had, in fact, collaborated with my very first PhD, my very first lab student, Parantu Shah, who joined our lab as an MSc student. And this was between the years 98 to 2000, exactly when the lab started. And even though he had moved on and he was doing his PhD in Pierre Bourque's lab, and then subsequently had just joined Lars Jensen's lab for a postdoc, we reached out to him and we collaborated. And whereas Lokesh will explain the meta-analysis that gave rise to better understanding of this seemingly non-functional serine protease as to why they are there. And Parantu, on the other hand, will very beautifully explain what happened in between. Between 1999 to the 2006, what were all the developments in the whole area, in the area of development of biology of Drosophila genomes, and how we had tried to exploit that sort of information. So this, however, is still part A of the second story, and part B is coming back to Parantu Shah's inputs in fact, as a part of our investigation, which is to worry about even the functional serine proteases, because the serine protease is still a very broad family, and within that you can have trypsins or thrombins or plasminogen activators or elastases, coagulation factors, and so on. And whereas their active site catalytic triad is preserved in and around their substrate binding site, there were residues that are different, and this seems to be important to dictate what type of serine proteases they are going to be. And this kind of fine-tunes the substrate specificity, as it were. And what Parantu Shah, together with Lokesh, had investigated was to exploit this property of the substrate binding residues and how they are different across these subfamilies. And they had gathered this information using literature survey and so on. And they put it through a new protocol that we proposed, which we called as a functional residue clustering. It's somewhat intuitive. You cluster the sequences based on only the functional residue information. And this will be presented by Parantu as a narration. And that is the second story. All right, so first story is to look for serine proteases in 2 different plant genomes. And the second was to worry about non-functional proteases as well as subfamily specific identification, right? And then whereas serine proteases are a broad family, and within that you can have 50 types, we actually were more interested in another type of serine proteases, namely the Prolyloligopeptidases. And in fact, Lokesh's work only had led us to think about Prolyloligopeptidases a little more. And this part of the journey actually happened just after Lokesh had wound up his work, looking not only for serine proteases in plant genomes, but he had actually looked for them in many different bacterial genomes. And in fact, we got very good response because different public domain databases started writing to us and asking us to present and include, they included our predictions into their databases. And this was a very interesting journey. However, we began to look at Prolyloligopeptidases more closely, and Swati is going to narrate this part of the story and say how, if we were to search for these Prolyloligopeptidases, which contain an accompanying other domain, which we call shown in violet here, which is the propeller domain, and what is shown in the green and yellow is nothing but the core catalytic domain. They always need to be together for it to perform its special Prolyloligopeptidase activity. All right. So, but then, as mentioned earlier in the sequence search story, Swati investigated and looked for Prolyloligopeptidases in different bacterial genomes available then, and she used cascade sequence searches. And the cascade sequence search, again, we had explained it before, and she very beautifully showed that the presence of the neighboring families and their sequences, in fact, do not cause confusion, but enable better coverage of the Prolyloligopeptidases themselves. All right. So this is going to be also presented by, I guess you are not able to see the movement of slides. So what I'm going to do is I will stop sharing and I'll come back to the screen sharing. I'm going to just rapidly take you through the slides. Are you able to see now? All right. Yes. So what I was trying to say that the serine proteases were searched in. Are you okay? So I was going to say that different families of types of serine proteases were either species specific, or sometimes they have higher extent of orthology. This will be explained by Lokesh very soon. And he also showed that the phylogenetic clusters seem to kind of propagate or provide the information on their domain architectures. We had discussed that before. And here, I was going to say in the second story, as pointed out by late Professor Veronica Rodriguez of TFR Mumbai, there are residues such as the catalytic triad, which is a textbook example of enzymes and catalytic activity. But in the Drosophila genome, for instance, there were appreciable amount of serine proteases which did not contain the catalytic triad residues, and still they were expressed. So the question really was, why are they present and what is their biological role? And to infer this, together with Parantu, we investigated and we performed a meta-analysis using a lot of new information that was getting available over the time. And Parantu has been my MSc student, and he was my very first student in the lab. Although his tenure was between 1998 to 2000, as you can see, we kept in touch and we reached out to him for collaboration. And he, in turn, permitted us to collaborate with Pierre Bourque and Lars Jensen, from whom we obtained high throughput data. And all this was then contained and helped us to understand better about these non-functional serine proteases, as to why they are there, what they may be doing in the cell, etc. All right. And the second part of the second story is to ask, even for the functional serine proteases, in and around the catalytic triad, depending on what type of subfamilies they are in, whether they're trypsins or thrombins or plasminogen activators or elastases and so on, there could be fine-tuned substrate specificity differences. And these were driven by an idea that is going to be presented by Parantu, which we call it as functional residue clustering. Then the term is very intuitive. You look for functional residues and use them alone for the clustering and phylogeny, so that we can begin to appreciate what sort of subfamilies they may be present. So then the third story that's going to be presented today is on not trypsin serine proteases, but Prolyloligopeptidases, which we became interested because of Lokesh's work on the on searching for them and other types in plant genomes and in bacterial genomes. And Swati is going to present this work, and she in fact joined just after Lokesh left. And the Prolyloligopeptidases are very beautiful structures. They always seem to be coexisting with this sort of a propeller domain, which is shown in purple, and the catalytic domain is shown in green and yellow. And whereas we had searched for them in bacterial genomes, Swati is going to also explain in her narration how the presence of neighboring families matter. And in fact, they seem to help and not cause confusion or problem, because the presence of neighboring families and their sequences seem to help better coverage. And this has also been published in this paper in PLOS 1 by us. And then finally, we are going to have the fourth story, which is related to the third, but somewhat distinct, because Saumya, who's currently a PhD student who's continuing this work on the POPs, is going to show how it is relevant and hopefully important to look for POPs in bacterial genomes which are residing in the human gut, namely in the gut microbiome. So with this, I will stop, and I will request Shaya and Shafi to kind of coordinate and invite the presenters. Thank you.
So we would now like to invite Dr. Lokesh Tripathi to present his experience serving serine proteases in 2 plant genomes, as I mentioned, and other several bacterial genomes. Soon after that, he will also present a meta-analysis which enabled better understanding of non-functional serine proteases in Drosophila genome. Over to you, Dr. Lokesh.
Thanks very much, Shailya, and thanks very much, Sadamni, for inviting me. It's been a while, and it's a pleasure to be a part of CAPS once again, even if as a panelist. Thank you very much. And can I share my slides? Actually, is it? Yeah. Okay. So actually, I should apologize in advance. This is a work that this was carried out almost 15 years ago, and I'm looking back at it, actually led to a wave of nostalgia. I mean, things we had done, and I realized that how many of those approaches still remain relevant. We still talk about integrated multifold approaches back then, and now we call them multi-omics approaches. So it seems that science is never ending. It's progressive, it's cascading, to put a pun on it. And we build upon what we learned before, and we use this procedure now. Even now in my research career, so many years after leaving Mini Lab, Sadamni Lab, I still use multifold approaches in my research by combining data from different types. And I should thank Sadamni, because that's where the idea came from. I mean, what I learned in Sadamni Lab way back in my PhD, I'm still applying those principles even now. So without much ado, I think rather not go off my soapbox, I'll come to the presentation. So let me share the slides if I can. Yeah, it's fine. Yeah, thank you. So it will be a brief presentation. My name is Lokesh. Presently, I'm a research scientist in the Laboratory of Transcriptome Technology at Riken Center for Integrated Medical Sciences in Yokohama, Japan. So it's also correlates well with the title of my talk, which is Genome-wide Survey of Serine Proteases. This also happened to be the title of my PhD dissertation. So I've repurposed the same slides from back then. I think they've stood well to time, and it's possible I may be a bit rusty on the details. So please forgive me for some oversight, and please feel free to correct me anytime. So the reason, as Sadamni mentioned, the reason we picked up this very interesting family of serine proteases is because this is a very diverse group of enzymes which are involved in every imaginable process that could be linked with cellular physiology. In humans, they've been linked with tissue remodeling and coagulation. In other organisms, including humans, Drosophila, yeast, bacteria, archaea, they've been linked with apoptosis, with metabolism, with defense, with regeneration and reproduction, and even development. So therefore, it makes sense to understand these proteins in very in depth to understand what exactly are doing within the cell, because especially I think this holds true even now, but when we when we started, the in vivo substrates of these proteins were not characterized at all. I think still now, based on the very brief literature survey that I carried out, many of these proteins, we still don't know which proteins they target. Even with the availability of high throughput RNA-seq and other data, it's still challenging, but it's a non-trivial task to understand what these proteins are doing in the cell. So the idea was that in this process, I took up the genomes of human Arabidopsis and rice, as Sadamni very nicely explained, that there are two model species representing monocots and dicotyledons, the two main lineages in higher plants, and then using a series of sequence search methods, which I'm sure have been superseded by newer and advanced methods, especially HMM, which is now much faster than it was back then. It could take ages to run an HMM scan. Now I think Hammer 3 is quite fast, which is good. And then we used, once we obtain a list of putative or tentative serine proteases, we would filter them for redundancy, removing those which could be truncated fragments or perhaps just genomic noise, to obtain a non-redundant data set. Once we had this data set in hand, we went to work by looking at different features that were contained in these sequences, by looking at the alignments to different protein families, by looking at signal-peptide relationships, by looking at the chromosomal mapping and location, by understanding their homologous and orthologous relationships, by performing multiple sequence alignments and assembling phylogenetic trees to understand the distribution, evolutionary relationships, and could they be used to infer functional relationships among these proteins. So this is an example of the diversity of the serine protease family within plants or across the lineages. This work also drew in from later analysis that we published just before I submitted my PhD dissertation. So as you can see, that some domain architectures, each of these represents a protein domain family, but with independent and self-sustained function. So many of these eukaryotic proteins, they carry multiple protein domains, which comes as a combination of evolution and recombination with other proteins. And the more protein domains you have in a family, the more complex and more likely additional functions they are likely to have. Therefore, as we can see in this particular slide, there are some protein domains families, like the simplest trypsin family, which only contains a single proteolytic domain. It's reasonably present both in plants and both the plant species. And you also find them in every species that has been examined so far, from bacteria to archaea to eukaryotes. But then there are some proteins or some members of this family which contain an additional PDZ domain, which were found only in bacteria and eukaryotic species, but they were absent from the archaeal species examined back then. And of course, there are there are examples of subtlelicin family, which happens to be the largest family in all the plant species. And a particular domain combination, proteins containing this particular domain architecture, which is a pre-subtlelicin domain, the subtlelicin domain, and the P domain, they are the most abundant in both the species, and they were again excluded from archaea. But of course, we also found some more complicated combinations or more complex associations of domains, which were very specific in rice and not found in Arabidopsis either. So this leads us to believe that phylogenetic association, which is confirmed by phylogenetic association, that proteins containing particular domain combinations, they were established very early in evolution, and they were disseminated through species. As we can see that the clade 1 contains a single subtletin domain, but there are clade 2 which contains multiple subtletin domains over here. And eventually, sometimes these differences percolate down not just to the domain families, but also at the individual residue level. This is a very interesting family of signal peptidases, which we found that there were two broad categories of this family of these proteins in plants. One of them had, as you can see, there's a clear distinction in having a lysine residue in his. Basically, these two amino acid residues. This is the active site, which was highly conserved over here. You can see the arginine residue is highly conserved. You have IVL substitutions as well, isoleucine, valine, leucine substitutions. But the most interesting aspect was the presence of. You can see this difference over here. The residue which. The amino acids, the proteins which contained, which were clearly divided into two clades based on just these two amino acid substitutions, clearly suggesting that it's not just the domain architecture, but the catalytic activity itself, which is established very early in evolution and then disseminated down the species. So the idea is that once these different catalytic lineages, or so I would say proteolytic lineages were established, they go on perpetuating, multiplying in number and complexity as they need to perform multiple functions in the same domain. So this slide, so what we concluded was that the function clusters employing slightly varied catalytic mechanisms, they established early in evolution, and they've subsequently retained, possibly for adaptation to different physiological processes. Then once we observed these observations in plant species, we thought that how important are the functional residues? And if functional residues are actually diversified early in evolution, what happens if the functional residues are lost, or they maybe are mutated so such that the proteins cannot be deemed to carry out the initially envisaged roles for them. So in this case, we started looking at Drosophila serine proteases, which, as Sadamani mentioned, had a large chunk of proteins where the catalytic residues were mutated early in evolution, and therefore they were very likely not to carry out any evolution. But the question is, if these proteins are not performing catalysis, what are they doing here? They just can't be, it'll be immensely wasteful for the cell to express and produce the proteins in large quantities when they seemingly cannot catalyze. So one of the ideas was that they may not retain the ability to catalyze, but they will definitely retain the ability to recognize and bind to the substrate. So they may actually perform additional roles based on the additional domain combinations that exist. So our idea was to look at the enhanced functional repertoire of these proteins by looking at different data types and see if we can actually apply these principles to annotate different multi-member gene families which exhibit similar phenomena, which is actually a very common phenomena now. More lately, it was observed in kinases and many other genome families as well. So in this case, we employed a very sort of a comprehensive approach using multiple data types. So what we found was that initially, most eukaryotic proteins, they belong to large gene families. They have multiple domains. They participate in many cellular processes, and they're located in different parts of the cells. Therefore, the idea is that if you want to analyze them, understand them, you cannot rely on data that only examines one of these aspects. You have to look at data in its entirety, see if you can actually establish links with all these aspects, and most of these data are contained in different data types. So the idea is that if you leverage biological information from multiple data types, this is likely to be more robust and accurate in inferring functional associations. And this has to be done in a multi-omics framework. It's now known as multi-omics approach to biology these days. But back then we didn't use the term multi-omics that much. So in this case, that time gene ontology was still the go-to repository for annotating gene function. Now we have many other pathway databases, but they were much in their infancy back then. So we still relied on gene ontology. And as I think all of you would be knowing, you are fully aware that Gene Ontology Consortium annotates function using 3 different components. There is molecular function that's based on, say, whether a protein is a kinase or a protease or something similar, or what kind of property does it have, what kind of domain properties does it have. Second is the cellular process. This pertains to what biological processes has the protein been observed to involved in, or is likely to be involved in based on inferred associations. And finally, where in the cell this a gene or a protein is likely to function. It's a combination of these 3 elements together, which tells us more about the protein domain families and exactly help us pinpoint the precise functions of these proteins. So then the question comes that how do we go about integrating this information, and not just from gene ontology, but from other resources as well. So we coupled our expertise with sequence and structural analysis with using more metagenome or high genome or high throughput data in this kind of a framework. So we were quite comfortable initially we were using sequence and structural domain families to get information on these proteins. We coupled them with information from network analysis databases, which recorded protein-protein interactions or other physical interactions of these proteins with other proteins in the genome or the proteome, and looking at the disease membership of these proteins as well. And based on this information, based on the sequence structure information, we came up with this concept called functional residue clustering, where the idea was that the catalytically active, or those residues which are deemed to be functionally important, should be sufficient to assign some sort of a broad definition of broad functional category to most of the proteins in this gene family. I think Parantu's talk will tell more, will elaborate more on it, so I won't go into this in detail. But then the idea is, if you merge this sequence structure-based association with the more detailed with the high throughput genomics data, such as microarrays and protein-protein interactions, coupled with subcellular localization, coupled with the RNAi experiments, which were becoming more and more common at that time, then you are able to enhance or elaborate on the known domains of molecular function, biological process, and subcellular localization, and also help to validate the predictions that we are making. This is one of the questions that most of the reviewers always come up with, that, okay, fine, you've made a hypothesis, but how do you know this is true? So sometimes RNAi and other experiments do help to convince not just the reviewers, but also sometimes your own colleagues who feel that, yeah, okay, fine, let's take it up for the investigation. These proteins do offer, these genes do offer a lot of promises. So this is a schematic of the gene expression meta-analysis of all the serine proteases in Drosophila development. So here, this is the heat map of all the 204 serine proteases and their pathway memberships in Drosophila. The green space represents the time points where these proteins where the genes had no expression, or they were underexpressed, or not their expression was not detected. And the yellow regions represent those regions where the expression was much higher.
Not there. Here
I'm sorry.
Please go on.
Okay. So what we hear is, what we clearly see is that it's towards the later part of development. That means when the development stages reach their end, from embryonic stage to the later stage of Drosophila development, that most of these proteases become active. This makes sense. I mean, there are some involved in the initial stages, but most of them get involved in the later stages of development, which makes sense because it's the time when all the other protein families and all the other existing proteins have to be degraded, recycled, and a new set of genes take over. So the existing proteins have to be leave their function done and dusted. So they have to be put back into the recycle pool for the amino acids to be incorporated into new proteins. So that's where many of these proteins are involved. So as you can see, many of these proteins are involved in immune response, in development and patterning, in toll pathway signaling, in transport and digestion. And that's where we got our first clue for some of the very interesting proteins, for which the functions were not known. So we had the list of proteins which are involved both in toll signaling and development, namely Easter snake and GD. And then we found there was a very interesting protein which was tucked in between these proteins with very similar expression patterns, but for which we had no idea what it was doing there. And based on these assumptions, we actually concluded that CG3066 must be an important protein because its co-clustering is co-expressed with both Easter snake and GD, and therefore this protein must play a very important role in development. I'm sorry I didn't put the data here, but this protein actually turned out to be another very important player in toll signaling pathway together with Easter. So there are physical interactions involved as well. So once we had the gene expression data, the idea was that, can we also look at them at this is at the gene expression level. What about the protein level? So in this case, we examined the protein-protein interaction data for them, and we realized that many of these proteins interact with proteins which participate in signaling, defense, development, metabolism, gene expression, and transport. So this is a very diverse category. So based on the guilty by association principle that the genes which are involved in similar processes are likely to physically interact with each other, we were able to associate functions for a large number of proteins with them. And interestingly, many of those non-functional or semi-functional serine proteases, which we call serine protease homologs or serine protease-like proteins, they were found to have interactions with all the proteins in different categories based on Y2H data. This compared to very few in the existing data from string, which clearly suggests that using multiple data types or multiple data sources can actually boost the level of annotations you can assign. So what we found out was that based on literature, gene expression, and PPI interaction, about 41% of Drosophila SPs, that's a huge number for a multi-member gene family, they are associated with immune response. And this included about 21 genes or 21 proteins which were implicated in immune response against bacterial, fungal, and parasite infections. This goes on to show that these proteins, they form, if they are present in such large numbers in bacteria and also in other species, they are very much important for immune response development. And therefore, any therapeutic interventions, especially in the case of human clinical trials or human clinical studies, they need to leverage the role of these proteins in developing new therapies, which can actually help overcome drug resistance, among other things, which is a big issue in drug discovery these days. And most interestingly, for our purpose, about 27 of these serine protease homologs, or serine protease-like proteins, about 47%, were involved in immune response. And based on the RNAi screen that we also examined, about 10 of them were verified that, yes, they are involved in immune response. So what it told us that compared to the regular serine proteases, there was a specific recruitment of these serine protease homologs in immune response based on Fisher's test. And about 17% of those serine proteases, 100% of those serine proteases, which actually contain the CLIP domain, they all function in immune response. So based on combining the data from different sources, we could clearly assign a new role, completely new role for these seemingly inactive enzymes. Therefore, just having lost the capability to catalyze reactions does not make them functionally inert proteins. They tend to adapt. Nature is far too actually, I was frugal or conservative to let go of such waste opportunity. So what these proteins are doing is they are repurposed, or they are being repurposed for a different functional role, which retains some aspects of their serine proteolysis. That means they're able to bind and recognize substrates, but instead of cleaving, they sequester them, or they target them, or they sort of guide them to the eventual signaling pathway of eventual signaling processes that are involved in overall in the cell. And again, what we saw was that if you look at the cellular localization, much of these proteins were secreted, which is understandable because that fits in with what is already known. But also there was a good percentage of those involved with associated with mitochondria, associated with the membrane, and also those which were among other locations like nucleus and others. And in this case, we found a protein called CG8464, which was a mitochondria localized protein. And its human ortholog was known to be a mitochondria localized protease with pro-apoptotic function. But at that time, this was not known. And actually, the very year when we published this paper, actually, we submitted much earlier. It got published in 2008. But soon after our study was published, there came another study where CG8464 was actually shown to be a pro-apoptotic protein in this study in cell differentiation in 2008. So it clearly showed that if you performing a broad-based multi-omics analysis can shed much more information and can shed more insights into protein function. And therefore, this is an approach to be pursued, not just for examining protein domain families, but for examining any kind of biological data set in general. And this is to summarize these findings, that using different kinds of approaches, how many annotations were there and how many of them overlap with each other, and why it was necessary to take all of them into account instead of only looking at one particular interaction. As you can see, that many of these information sources are rather discrete. They provide an information that does not really overlap with other domains. For example, PPIs have only partial overlap with other data types. Gene expression itself shows some overlap with other things, but there are also a large number of specific domain-specific information. So therefore, to get a very holistic or a broad picture, you need to combine or merge all of them and then filter them for redundancy and reliability to pick up the best matching candidates and also to pick up the best source of information. So to summarize, we showed that using multifold approaches, about functional associations were established for 190 serine proteases in Drosophila. And this was a 10-fold increase in the pre-existing annotations. Large number of catalytic active and SPHs were judged to be associated with development and immune response. That was a novel discovery, and which seemed to be getting validated as more and more studies papers have come out. I haven't summarized them here, but I did see a few of them. And finally, data integration is an effective approach for reliable function annotations of genes and networks. So with that, I would like to acknowledge Saudamini for all her help and assistance and for patience during PhD. I remember sometimes she used to feel like pulling her hair out with my seemingly lack of many things, but she was persistent, she was patient, and she actually helped me through a lot of them. Parantu, who was a former student, but actually came back to collaborate with us, and I learned a lot from him and from his colleagues, and also all the former members of RS Lab at that time, and who were actually we worked together as a family, and that was something I do tell people wherever I've been to, that Saudhamani Lab was one of those where you never felt out of place. You're part of a family, and long may it stay. Long may it stay. And I hope it's the same right now. So with that, I'd like to thank you for your attention, and I'll be happy to take any questions.
Thank you, Dr. Lokesh, for sharing your science and philosophy of science. We'll be having question at the end in the question answer session.
Yeah.
And so now we'll present the narration by Dr. Parantu, where he explains how functional residue clustering enables association to subfamilies. I hope my slides are visible and it's audible to you.
Student of Dr. R. Saudhamani at the CAPS lab at NCBS. I am currently an Associate Director of Bioinformatics at EMD Serrano, otherwise also known as Merck KGaA, outside of US and Canada. Today, I plan to talk about functional residue clustering for substrate specificity transfer, a case study that we did in genome-wide function annotation. So, there are more than 200 serine proteases-like domains encoded in Drosophila genome. And when I say we did functional annotation, we provided molecular function, biological processes, and subcellular localization to as many as possible of those Drosophila genes. This work was done with Lokesh Tripathi of Saudhamani's lab, as well as members of the Borg group and Kevin White's lab. However, to start with, let me tell you a story. Stories are always better than the dry work. The project actually started in 2001 when Dr. Veronica Rodriguez actually asked us to provide some understanding of function of a gene called masquerade. The gene encodes for a secreted molecule with a serine protease-like domain, which is required for somatic muscle attachment. However, something that was interesting is that the domain would lack the catalytic activity due to a key change in the catalytic triad residue. We successfully finished the work and published the paper in 2008. However, in the meanwhile, the field of genomics has completely changed the way we worked. So for example, in 2000, the euchromatic genome sequence of Drosophila melanogaster came out, which allowed us to expand the work from one serine proteases to the entire genome. We got help from the microarray-based whole genome gene expression map of Drosophila life cycle done by my postdoctoral advisor in Dr. Kevin White's lab. We had string database of protein-protein interactions, and that was published in 2005, which was done by Pierre Borg's group, where I did my PhD. There was genome-wide RNAi screen for Drosophila immunity, as well as the IHOPE literature server that provided information on gene and proteins, done in Dr. Alfonso Valencia's lab, who was my PhD supervisor. These advances made function annotation using data mining on a large scale possible. Today, I'm going to abridge that study and just talk about molecular function, transfer of substrate specificity, or a relationship that you can hypothesize between an active protease or a protease-like domain because of the mutations in catalytic triad. So we utilized PDB, the source of three-dimensional structures, SCOP, the classification of proteins that would allow you to understand the evolutionary relationships between those proteins which has structures determined, and SwissProt, which would provide us genomic sequences and annotations, which we could in essence use in a form of a multiple sequence alignment. So we derived information from protein inhibitor complexes, trying to really understand where inhibitor binds to those proteases. And some of those residues would be involved in the specificity of binding or the substrate specificity for the proteases. SCOP provided us non-redundant information and structural base alignment that we could do in order to refine the multiple sequence alignments. Swissprot would allow us lots of sequences where we can understand about the subfamilies and positions that are actually conserved across those subfamilies. We extracted those residues involved in substrate binding and catalytic specificity, extracted the sequence positions from there, and ran a hierarchical clustering in order to really understand the similarities between the catalytic sites and binding specificity sites of those proteases. Then we could actually use this relationship expressed as correlation coefficients to really understand or transfer specificity between protease that has known sequence or structure annotated in the databases to a novel protease. We could also create hypotheses about putative competitive inhibitors where proteases are very similar to a protease-like domain, but that are not functional. So here is the detailed example. For the trypsin-like serine proteases, we actually utilize the superfamily level structures for which there are basically 35 proteins right now, as in 2021. We use a little bit of less. We analyze the protein and inhibitor interactions and found that different families of serine proteases, trypsin, thrombin, coagulation factors, and so on, indeed interacts with their inhibitors at different residues. And then we try to really understand which of these residues, in terms of a sequence alignment adjusted by structures, where there is a subfamily specific conservation, or there is a difference, trying to really get us a lot of information about the evolution, structure, and sequence. So here is the example of the output. And here is a substructure of that output. And what we can basically see in the tables 1 and tables 2 is in really you could actually take a structure, basically where everything is known, its function is known, and you can actually translate it to Drosophila serine proteases. Similarly, like I mentioned, that you can have a serine protease-like domain correlated to serine proteases, and really try to make the competitive inhibitor relationships. So for example, masquerade to CG5246 in the Drosophila genome. We evaluated the performance of substrate specificity transfer, where we have known evidences from literature. At the correlation coefficients of 0.3, the specificity of method in transferring the substrate specificity was 0.86, and the sensitivity was 0.98, which was a very, very high quality. So thank you very much, everyone, and I'm looking forward to your comments. Have a good day.
That's about serine proteases stories. Now we will move to the next set of stories. Dr. Shwati Kaushik will present our experience in searching for Prolyloligopeptidases in bacterial genomes using cascade sequence searches.
To improve coverage of remote homologue detection. And in second part of my talk, I will discuss how we systematically mine bacterial genomes to understand diversity and functional role of prolyloligopeptidases in bacterial genomes. Prolyloligopeptidases are very unique serine proteases. They can only cleave peptides which are smaller than 30 amino acids in length. So they are highly selective and very specific towards their substrate. Prolyloligopeptidases are widely distributed in all domains of life, ranging from bacteria to archaea to plants and humans. Prolyloligopeptidases are given as oral supplements to the patients with celiac disease, which is gluten sensitivity, as these enzymes can degrade gluten very specifically. On the right side of the slide, I'm showing the structure of prolyloligopeptidases, which consists of two domains. On the top, you see is alpha-beta hydrolase domain, which has active site. And on the bottom, you see is a beta propeller domain. Homologous relationships can be broadly divided into two groups, close homologues versus remote homologues. So close homologues are the ones where sequence and structural similarity is pretty high and they can be easily detected. While remote homologues are the proteins where there is very low sequence similarity, usually less than 30%, but they have high structural similarity. And detection of such remote homologues between two proteins, we can use intermediate protein sequences as shown over here. Such intermediate sequences, for example, protein C here, they share sequence properties of both the protein A and B, and that's why it can be used to detect remote relationships between two proteins. And these methods have efficiency of 70% over simple pairwise method. One of the very conventional method for homology detection is SciBlast, which has been used very frequently. I'm sure like many people have used in the audience also. So Sandhya Sankaran from Sadamini's lab had developed a more rigorous approach of remote homologue detection by performing multiple rounds of SciBlast, which we call as cascade SciBlast. So this involves not only using just one time running of SciBlast, but we also go beyond just doing first generation. We run SciBlast to second and third generation to detect more and more remote relationships, not only at family level, but also at superfamily and full level. So in my PhD, I've extended this protocol by employment of multiple queries of a family into rigorous cascade SciBlast searches to predict distant relationships of Prolyloligopeptidases from plants. So what you see here is a neighbor joining tree of pop domains from Arabidopsis in rice, and this was work by Lokesh in our lab. So the question that we ask is, instead of using just one query sequence, what happens if we use all of these sequences as query one by one? And we wanted to see how the coverage is affected by these individual sequences of a family. So in this graph, I'm showing how coverage varies when each member of POP family was used as a query on x-axis. So these numbers are basically A represents Arabidopsis POPs and O are rice POPs. And on the y-axis, I'm showing the coverage at family level. On the right side of this, on the left side of this plot, the sequences or the queries are showing almost like 40 to 50% of the coverage at family level, which is considered as low. But on the right side, you see some of these sequences are reaching a coverage of 80% at family level, which is pretty good. In general, SciBlast coverage was relatively poor as compared to cascade SciBlasts. This work is also showing that different queries behave so differently in these sequence searches. That's why picking the best query before you start any sequence search is very important. So we ran this approach on multiple other protein families from SCOP database, and we found that Cascade SciBlast shows really good coverage at family, superfamily, and full level with little loss of in precision. So I'm going to show you in one example in detail, showing the importance of neighboring families of a query sequence. So POP belongs to hydrolase superfamily, which has 41 families, which are shown in blue color over here. Let's say we have one query sequence from the POP family. And when we do first generation searches, we can pick only some members of alpha beta hydrolase superfamily, shown over here in green. But when we go beyond first generation to second generation and third generation, we can pick many more members of this alpha beta hydrolase superfamily, which were not picked with just one generation. So this shows how neighboring families of a superfamily can play a very important role in detection of distant relationships, which we can easily miss. In summary, our work signifies the importance of using multiple query sequences and the role of neighboring families in remote homology detection. Now I'm going to switch gears and discuss how we have used all the sequenced bacterial genomes to understand diversity and evolution of Prolyloligopeptidases in bacterial lineages. So we use various sequence search approaches to mine almost 1,200 bacterial genomes for which genomes were sequenced at that time. Which are broadly categorized into 23 bacterial and 4 archaeal phyla. And from that, we have picked almost 3,000 POP homologs. And we found that POPs are widely distributed not only in bacterial but also in archaeal lineages. Using phylogenetic clustering analysis of these POPs, we found there are 9 distinct clusters as shown over here. And these 9 clusters, interestingly, had co-clustering among members of different phyla, as shown by different colors over here, which shows that there is high conservation of POPs with no species-specific function within bacterial lineages. We mine these distinct clusters to understand like what if there are some specific motives that we can identify, like which are making them to cluster into 9 distinct clusters. So we identified that there are many class-specific motifs in these group of Prolyloligopeptidases. And on the basis of those class-specific motifs, we proposed a classification of bacterial POPs into 8 distinct types. And then we also did a more detailed analysis and found that some of these motifs were surface exposed, which could be responsible for protein-protein interactions with other interacting partners, while some of the motifs were present in the core of Prolyloligopeptidases. So overall, these subtypes also differ with respect to some of the functionally important residues, which can lead to possible differences in the substrate specificities of Prolyloligopeptidases. In summary, our work shows the use of multiple query sequences and role of neighboring families to improve remote homolog detection. And today I didn't get a chance to talk about Cascade HMM, which is another efficient protocol that we developed using hidden Markov models for cascade searches. And in second part of my talk, I discuss how we have mined different bacterial genomes to understand diversity and functional role of Prolyloligopeptidases and identified many class-specific motifs and functionally important residues. So if you are interested in learning more about the work that I discussed today, here's a list of publications related to my talk today. And with that, I would really like to thank Professor Sudhamini for being such a great mentor. I really miss you, ma'am. And thanks to all the people involved in the work. And here's a picture of CAPS in 2011. So all of us are looking so young, and time flies. So thank you, everyone, and thanks again for giving me the opportunity to talk today. Thank you so much.
Yeah, so next we have Soumya with us. She will be sharing her experience continuing on this journey, searching for POPs in gut bacterial genomes. Over to you, Soumya.
Thank you, ma'am, for the opportunity, and good afternoon, everyone. So continuing in the previous work, we are now researching the POPs from the RIPSIC database, which is a non-redundant and well-analytic set of sequences, and we have taken the number of genomes which complete annotation and fully sequenced, and those comes to around 32,000. And from those genomes, if we search for the POP family protein, we found around 61,660 protein, which is distributed in those 32,000 genomes. So okay, so now in this figure, if we see the representation of different phyla in the FC complete genomes, we see that proteobacteria almost dominates, like is more than 50% of the total genomes, and followed by bacillomyta and actinomyta. So this is an ongoing work. We'll continue working on this, and And then coming to the next part of the story, that is the POPs in gut microbiota. And we saw how we know that the POP has the potential to use as a therapeutic for celiac disease. So celiac disease is an autoimmune disease. It is caused by the abnormal immune response in some genetically predisposed people. So there is this basically because when those people person ate gluten rich food, so this is because due to the peptide rich gluten are not break down properly in the gut. So there are many studies which shows that POP can be used as a supplementation, and so for example, POPs from Aeromonas pingomerata and Micrococcus xanthus has shown that it can lead to rapid cleavage of gluten peptide, but our focus in the study is to find potential therapeutic POP from the gut microbiota that could be used as that has the probiotic property, so it could be used as an oral therapeutic. So here the overview of the methodology we use. So very briefly, I'll just say that we have used proteomic assemblies from many different sources. For example, BioML, HumGut, HMP, and CGR. So all these studies combine different methods like all the culturable microbiomes plus the metagenomic studies. And we got around 10,000 genome, and then by doing the data mining and cleaning up, we got around 4,000 odd genomes. And then we look for the domain architecture. Like we know that POP has the classic 2 domain architecture of catalytic and the domain and other domain. And so we got around 734 odd POPs. And then we have done the blast search with the known template, which is the one template with salt crystal structure. Then after doing the search, we found that around 10 proteins which could be used as a modeling. Like we have used structural modeling, and then we tried to dock with the known potential epitopes for celiac disease. And from those we selected around 8 POPs from the 8 genomes, which has also the probiotic properties and also could use as a potential therapeutic agent. So coming to the distribution of the gut microbiota in the bacterial phyla, we see that gut microbiota is dominated by 4 types of phyla. So those bacteroids, actinomyces, firmicutes, and proteobacteria. So this graph shows the distribution of POPs in this 4 phyla. So we see that bacteroids almost dominates with 50%, almost around 50% of POPs found in the bacteroids phyla. And here in the right diagram graph, we see that most of the POPs from firmicutes phyla have only one or 2 POPs, but most of the bacteria from the bacteroids have more than 1 POP. Some of them also go to around 10. So if we see the selected POP species which has the probiotic properties, this is the list of the major species which I've selected, which have this classic 2 domain architecture and also belongs to the different phyla, which like some we have selected some from proteobacteria, some from bacteroides like this. So we have selected E. coli, which is a known probiotic, and also Prevotella, which is the most dominant species in the gut, and it is also considered as a next generation probiotic. And Bifidobacterium and Clostridium, they're very well known probiotics, so it has also immunomodulating capacity. And Faeculobacterium is and Bacillus subtilis, they are also used for treatment of some gastrointestinal disease like IBD and all. So these are the final selected set, and we'll also going from functional analysis in the next stage of our study. So with this, I thank ma'am and everyone. So any questions? Thank you.
Right. Yeah, so we have heard the four different stories. One was about searching for serine proteases, comparing them between 2 different plant genomes. We heard that from Lokesh, and then we went on to worrying about the seemingly non-functional serine proteases. And this was a journey started in 1999 and went on until 2008. And this was explained by both Lokesh and bit by Parantu, who talked about the history and the developments. And then we looked at the kind of subfamily specificity and annotations using functional residue clustering that was presented by Parantu. And then the third story was on the Prolyloligopeptidases, and especially doing sequence searches in different bacterial genomes using cascaded approaches, and how presence of neighbors and their sequences could in fact enable better coverage, and they don't come in the way. And finally, we also heard about our extension and a close look at gut bacterial genomes for the same. So I hope you enjoyed this set of narrations, our long-term journey, literally starting from 1998 until 2023. And people like Soumya are continuing, are present PhD students. But then in the first and second stories, we heard from Lokesh, and he's currently in Kyoto. He's a postdoctoral fellow there. In the National Institute of Biomedical Innovation. And we have also heard from Parantu, who's now an associate scientific director at MD Serrano. And then we heard the third story from Swati, and she's a research scientist at Gilead Sciences at San Francisco, and Soumya, who's with us now. Okay, so with this, I would stop, and then I invite Lokesh and Soumya to come online and take some of the questions that have come up. Yes, what are the questions?
Oh, yeah, I'm here. So, shall we start answering the questions now?
I think so, yeah. Yeah, so maybe Advait or Vikas can read out the questions and then we'll take it up.
Yeah.
So I'm at home, so you may hear these noises from time to time. My younger one is quite active.
So we have first question. What are serine protease homologs or SP-like protein in terms of function? So perhaps Lokesh can take this.
Okay.
Yeah.
So I think, do I have to click on answer live or type something? Okay, I'll click on answer live. Okay.
We can speak up live.
Yeah. So the thing is that as we discussed, serine protease homologs or serine protease-like proteins refer to those proteins which are sequentially very similar to serine proteases. They have the proteolytic domain. I mean, based on sequence and structure analysis, they seem to share a great deal of sequence identity with the known serine proteases. But it's believed that their catalytic function is compromised because the active site residues are either mutated or missing. So the idea is these proteins are present in the cell, but they do not function as proteases. But as we show that they may not be able to catalyze, but they retain the ability to recognize and bind substrates which are recognized by the fully functional serine proteases. So the hypothesis is that these proteins, they are involved as sequestrers, or they function as competitive inhibitors of the regular serine proteases. That means it's a regulatory mechanism where these proteins, they prevent the functional serine proteases from catalyzing or proteolytically cleaving. Sorry about that. Yeah. So these proteins, they prevent the active proteases from catalytically cleaving their substrates and thereby provide a very regulatory mechanism by which they can control or modulate the amount of the kinetics of the kinetics of proteolysis that happens within the cells. If the substrate is sequestered by these proteins, they cannot be cleaved by the regular proteins. So they can release these substrates in a very guided or a graded manner to regulate the rate of proteolysis. And also because many of these serine protease-like proteins carry additional domains. So they can perform roles in other processes like immune response or in metabolism, where those other functional domains can carry out these regions. But right now, the hypothesis is primarily they function as substrate sequestrers or competitive inhibitors.
Thank you, Lokesh.
Yeah. All right. So thanks, Lokesh. Now we will take next question, which is, with the cascade method of homology detection, are there a lot more false positives as compared to normal pairwise method? If so, how are they filtered out? Soumya can take this question.
Yeah. With cascade method of homology, Whereas if we go for cascade fibrillast, we don't see a lot of false positive there compared to normal pairwise method. But if we go for a different type of cascade method like cascade HMM or other, then we could see the false positive could increase a little bit. And if that is the case, then we can filter out like by looking at the domain architecture, and in that case we can filter out if that domain architecture is totally different and fold is totally different. So in that way we could filter it out.
Yeah, so just to thanks, Soumya. Just to continue on that, we also have additional filters such as query coverage that makes sure that we are not picking up suboptimal comparisons, and then also we keep each of these generations, we keep the e-value very, very stringent so that there is less chance to go on to false positives. And yeah, we don't encounter that too much.
Thank you so much. Thank you, ma'am. We have next question. Are POPs found in other commensals apart from those found in the gut?
Yes, so we know that POPs are also expressed in the brain, and as far as the other commensal in human body, we have also know that it expressed in the skin and mouth also. But in our study, where focus is the gut microbiota, because we want to have some potential therapeutic, which as a probiotic, which can be taken as oral therapeutic.
All right. Thank you so much. We have next question. How do you prevent false positive hits in cascade fibrillast, as these false positives can propagate across the generation of cascade search or pick up unrelated proteins in search? I think the second question has already addressed this. So we can move to next question, which is, can you please comment on the evolutionary relationship of serine protease family? I guess Lokesh can take this question.
Can you explain this again? This is the cascade method of homology detection. Serine protease evolutionary relationships. Okay. So I think the idea is that this is a very broad category. Serine proteases refer to any protein or any protease which is proteolytically active, but the only shared similarity between them is that they use serine as an active site residue. They can be completely different proteins belonging to different superfamilies and even different folds. For example, trypsin and cytolysine-like proteins, they all belong to different folds. So evolutionary relationships within the fold or within the superfamily can be quite close to each other, but across the fold, they are very diverse. They're completely different from each other. The only common case being that they use serine as an active site residue. So I think the hypothesis is that all these families, they emerged as a consequence of convergent evolution, where the nature realized that instead of duplicating the entire sequence, it's best to just repurpose the same catalytic arrangement in different protein folds and in different protein families. So you can say that there's no more similarity between a trypsin and a cytolysine than there is between a trypsin and a kinase.
Yeah, so yeah, thank you, Lokesh. In that sense, seems to be even worse than convergent evolution, because they seem to have a common biochemical function, that's it, and their folds are different. They definitely don't see each other in sequence searches, and some of them are membrane bound, some of them are not. So there's a lot of variety, and as mentioned, there could be as many as 50 different types of serine proteases, and we have presented our journey. And we have examined trypsin type serine proteases and the Prolyloligopeptidases in greater detail. But we would urge you to actually read up the paper by Lokesh and myself, where we have closely compared different types of serine proteases, not only between Arabidopsis and rice, but also within bacterial genomes. Thank you. All right. So I hope you have also enjoyed listening to this set of stories as much as we enjoyed narrating them. And thank you for the great questions. And thanks to the speakers who have come online and also shared your narrations as recordings. And we will see next week, but I would also request Shafi to present the relevant publications and then also announce the title for next week. Shafi, please.
Thank you very much, Adamni, for the invitation. It was a pleasure speaking to all of you. Maybe let's stay in touch and maybe have more correspondence, regular correspondence. Thank you very much, all of you, for your attention. See you. Bye-bye.
Third view of right from proteases POPs. First two are from Tripathi and Pansha et al. And the both are from Saudi Kaushik. And there are a few other publications you can see in our repositories. So for the next week in CAPS talk, we have some interesting topics. We have titled it "Needle in Haystack: Looking for Nucleotide Binders." So you'll be hearing about DNA binding proteins and RNA binding proteins. Please stay tuned.
Thank you. Thanks, everyone. Thanks to the team who have been helping us so much, and also to the communications office of NCBS, who has been tremendously helpful, also the instrumentation, and to the audience who have been patiently listening to our stories. And we do hope to see you all next week. Until then, take care. Bye-bye.